Hi, I'm Jason Fly, and I'm a graduate student at the University of Wisconsin-Stevens Point. And my project was made possible by the Friends of Sunset Zoo. So, let me show you my project. Welcome to Badlands National Park. The park is home to incredible geologic features and an array of ecosystem types. As a researcher in the park, I focus on the prairie dog colony ecosystem. Many animals use and rely on prairie dogs for prey and habitat. The endangered black-footed ferret takes prairie dog reliance to a whole other level. They entirely rely on prairie dogs for both food and shelter. So due to the reduction of contiguous prairie dog colonies by human means and the introduction of disease, black-footed ferret populations have crashed. My research focuses on the disease part of that equation. I study the plague. The plague is a non-native bacterial disease carried by fleas. Because it's not native to the United States, our native species are highly susceptible to it, including the prairie dog and the reliant black-footed ferret. So you might be wondering, what actually is my research project? Essentially, I'm a plague detective trying to solve plague mysteries. So take, for example, last year. Badlands had its first plague outbreak in 15 years. Take a look at this. So you got Colony A, Colony B. They're both plague positive. In reality, they're 20 miles apart. And there's a geographic feature between them. So as a scientist, there's two things we're interested in. Were they local plague outbreaks that were sparked by something occurring in the environment in each colony, like the climate, or was there a transmission event between the two? We're currently trying to study that first question. How do fleas move locally? To do that, I am trapping prairie dog family groups and picking the one that has the highest flea load. The one with the highest flea load, we are giving a fluorescent dye called rhodamine B. Fleas feeding off the dyed host will get a dose of that rhodamine B. And then we will retrap the prairie dog families over a series of days and collect all of their fleas. The collected fleas I will later analyze by fluorescent microscopy, where we can then detect flea movement between prairie dogs, which simulates how the plague is transmitted. I hope with this project to provide a framework for future disease researchers studying invertebrate vectors like fleas, ticks, flies, mosquitoes, or like midges. I want to thank the Friends of Sunset Zoo for helping fund my research.